Hello students, welcome back to Gray's Gyan. Today we're going to discuss intracellular proteolytic mechanisms. Your cells are constantly making proteins, using them, and sometimes damaging parts of themselves. If all this waste was never cleaned up, the cell would slowly fill up with junk and stop working properly. So have you ever thought about how your cell cleans up this mess? Our today's topic is intended to answer this question. We will understand 3 brilliant systems your body uses to handle this mess. The Autophagy Pathway, the Ubiquitin-Proteasome Pathway, and the Mitophagy Pathway. We'll also cover a brand new discovery that completely changed how we understand one of these systems. Let's get into it. Before we go deep into each pathway, let's get the big picture. Your cell has 3 main intracellular degradation systems, and each one has a different job. First, autophagy. This is the cell's bulk recycling system. When the cell is stressed, say, it's running low on nutrients, it wraps up damaged organelles and cytoplasmic content and digests them. It's literally the cell eating itself. In a good way. Most of the time. Second, the Ubiquitin-Proteasome Pathway. This is more targeted. It's like a molecular tag and destroy system. Proteins that are misfolded, or that have done their job and need to be removed, get tagged with a small molecule called ubiquitin, and then fed into a protein shredder called the 26S proteasome. Third, mitophagy. This one is specifically dedicated to damaged mitochondria. When a mitochondrion is beyond repair, 2 enzymes, PINK1 and Parkin, come together to mark it for destruction. Now, each of these is described in your Gray's Anatomy textbook, referenced in Figure 1.30. Let's go through them one by one. Here is Figure 1.30 from Gray's Anatomy, and this is your anchor diagram for this entire topic. Panel A shows you the autophagy pathway. You can see the phagophore forming, wrapping around the mitochondrion, becoming the autophagosome, and finally fusing with the lysosome. Panel B shows the Ubiquitin-Proteasome Pathway, the proteins being tagged, recognized by that barrel-shaped 26S proteasome, degraded, and then the ubiquitin being recycled. Panel C is mitophagy, PINK1 sitting on the mitochondrial membrane, recruiting Parkin, and initiating targeted destruction of that mitochondrion. This figure is originally from Kirzenbaum and Trace, Histology and Cell Biology, an Introduction to Pathology, 5th edition, Elsevier 2019. Keep this figure in your mind as we go through each pathway. Alright, let's talk autophagy. The word itself comes from Greek, autos meaning self, and phagian meaning to eat. So, self-eating. It begins with something called the phagophore, a curved membrane that comes from the endoplasmic reticulum, the Golgi, or even the plasma membrane. Now, 3 proteins are crucial here. ATG5 and ATG12, these are autophagy proteins that form a multimeric complex with ATG16L on the phagophore membrane. And LC3, light chain 3. This protein is inserted into the phagophore and it is the marker of autophagy. If you see LC3 in an experiment, you know autophagy is happening. Step 1. Phagophore formation. The membrane starts curving. ATG512 by ATG16L assembles on it. LC3 gets inserted. Step 2. Autophagosome. The phagophore expands, wraps around a piece of cytoplasm or an organelle, like a damaged mitochondrion, and completely seals it off. This creates a double membrane structure called the autophagosome. Step 3. Lysosome fusion. A lysosome fuses with the autophagosome. At this point, the ATG512 by ATG16L complex is recycled. But LC3? LC3 stays. It remains associated with the double membrane. Step 4. Autolysosome. After fusion, the structure becomes an autolysosome. And here's a key exam fact. The autolysosome has a single membrane, not double. The lysosomal hydrolytic enzymes break down everything inside. The end result? Amino acids, lipids, sugars, all recycled back to the cytoplasm. One more important thing, autophagy is cytoprotective. It helps the cell survive stress. But when stress is extreme and prolonged, autophagy can actually precede apoptosis, cell death, and progressive dysfunction of autophagy over time. That's closely linked to aging. Now let's move to the Ubiquitin-Proteasome Pathway, arguably the most important protein quality control system in the cell. Here, we're not dealing with whole organelles. We're dealing with individual proteins, specifically those that are misfolded, abnormally assembled, or have already completed their function and need to be removed. The process starts with ubiquitin, a tiny, highly conserved protein that acts as a molecular tag. There are 3 enzymes involved in attaching ubiquitin to the target protein. E1, ubiquitin activating enzyme. This activates the ubiquitin molecule in an ADP-dependent step. E2, ubiquitin conjugating enzyme. This carries the activated ubiquitin. E3, ubiquitin protein ligase. This is the one that actually transfers ubiquitin onto the target protein. And importantly, E3 determines specificity. It decides which protein gets tagged. This process repeats, building a polyubiquitin chain on the target protein. Now the polyubiquitin tagged protein is recognized by the 26S proteasome. Let's look at the structure of this proteasome, and this comes directly from Tomko and Hochstrasser, Annual Review of Biochemistry, 2013. They describe the proteasome as having 219S polar regulatory caps, at the top and bottom. 120S central barrel. This is the actual proteolytic chamber. The 19S caps are responsible for recognizing the polyubiquitin tag, unfolding the protein in an ADP-dependent manner, and threading it into the barrel. The 20S barrel chops the protein into short peptide fragments. Then finally, step 4, the polyubiquitin chain is disassembled, and the ubiquitin monomers are recycled for future use. What kinds of proteins does the proteasome degrade? Great question. Cyclins, to control the cell cycle. Misfolded or abnormally assembled proteins. Transcription regulators. Immune response mediators. It also participates in apoptosis and cell cycle arrest. The 26S proteasome is found in both the cytosol and the nucleus. And now, this is the part I'm really excited to tell you about. Because this is brand new science, published in Nature, March 2025. The paper is Cell Autonomous Innate Immunity by Proteasome-Derived Defense Peptides by Karen Goldberg, Arseny Lobov, Paula Antonello, and colleagues from the Weizmann Institute of Science. For decades, we believed the main job of proteasome-derived peptides was antigen presentation, displaying bits of proteins on MHC class I molecules for T cells. But Goldberg and her team discovered something completely new. When they tracked how the proteasome's activity changed during bacterial infection, they noticed something unexpected. The proteasome's peptide cutting pattern changed. It started producing a specific class of peptides during infection. And when they looked at what those peptides were, they matched the sequences of known antimicrobial peptides. Peptides that physically disrupt bacterial membranes and kill bacteria. This means the proteasome is not just a protein shredder, it's also a natural antibiotic factory. They called these PDDPs, proteasome-derived defense peptides. And here's the staggering part. When they used an algorithm to analyze the entire human proteome, they found that 92% of human proteins contain hidden antimicrobial peptide sequences within them. During infection, a protein called PSME3 is recruited to the proteasome, which increases the tryptic-like cleavage, essentially unlocking the antibacterial mode. This has enormous implications for antibiotic resistance, for innate immunity, and for how we understand the proteasome. Gray's Anatomy has already cited this paper, which tells you just how significant this discovery is. Now let's talk about mitophagy, the most targeted of the 3 pathways. While autophagy is a bulk process, and the Ubiquitin-Proteasome Pathway handles individual proteins, mitophagy is specifically about damaged mitochondria. Why is this important? Because mitochondria are critical for energy production. But they can get damaged by reactive oxygen species, by mutations, by cellular stress. and a damaged mitochondrion is dangerous. It can trigger apoptosis. So the cell needs to eliminate it quickly. Two key players. PINK1, PTN-induced putative kinase 1. This is a protein kinase that sits on the outer mitochondrial membrane. Under normal conditions, PINK1 is rapidly degraded. But when a mitochondrion is damaged, the membrane potential is lost, and PINK1 accumulates on the outer membrane. Parkin, a ubiquitin ligase. Similar in function to E3 in the Ubiquitin-Proteasome Pathway. Normally, Parkin sits inactive in the cytoplasm. But when PINK1 accumulates, it recruits Parkin. Step 1. PINK1 accumulates on the damaged mitochondrion and recruits Parkin. Step 2. A Parkin-PINK1 complex forms on the mitochondrial surface. PINK1 phosphorylates and activates Parkin. Step 3. Activated Parkin transfers a polyubiquitin chain to proteins on the outer mitochondrial membrane. The 26S proteasome is then recruited, recognizes these tags, and degrades the tagged proteins, ultimately leading to the destruction of the entire mitochondrion. Key point to remember: in mitophagy, no autophagosome is assembled. This is the critical difference from the autophagy pathway. Mitophagy goes directly through the ubiquitin-proteasome machinery. Now, here's the clinical bombshell. If PINK1 or Parkin are mutated, the complex doesn't form. Dysfunctional mitochondria accumulate. And this leads to one of the most important neurodegenerative diseases we know, Parkinson's disease. This is why mutations in the PINK1 gene and the Parkin gene are among the known genetic causes of familial Parkinson's disease. Let's take a moment to consolidate what we know about the 26S proteasome, because this structure comes up constantly in exams and in clinical contexts. Based on the work of Tomko and Hochstrasser, 2013, here's the structure clearly. The 19S caps. There are two, one at each end. These are the regulatory subunits. They recognize the polyubiquitin tag, unfold the target protein using ADP, and feed it into the barrel. The 20S barrel, also called the core particle. This is where proteolysis actually happens. It has an inner proteolytic chamber with active sites that cleave proteins. Together, 19S plus 20S plus 19S equals the 26S proteasome. And to be precise, the 26S proteasome is a multicatalytic protease. It has multiple types of active sites that cleave peptide bonds in different ways. It's found in both the cytosol and the nucleus, so it can degrade proteins in either compartment. Functions at a glance: removes misfolded proteins, degrades cyclins for cell cycle control, processes transcription regulators, participates in immune responses via MHC class I antigen presentation, and, as of 2025, generates antimicrobial defense peptides. Perfect. Let's now do a rapid comparison of all three pathways, because this is exactly what gets asked in exams. Target: autophagy, whole organelles and bulk cytoplasm. Ubiquitin-proteasome, individual proteins. Mitophagy, damaged mitochondria only. Signal: autophagy, nutrient stress or cellular damage. Ubiquitin-proteasome, polyubiquitin chain tagging. Mitophagy, PINK1 activation, Parkin recruitment. Autophagosome formed? Autophagy, yes. Ubiquitin-proteasome, NO. Mitophagy, NO. Key structure: autophagy, autolysosome. Ubiquitin-proteasome, 26S proteasome. Mitophagy, 26S proteasome via Parkin. End product: autophagy, amino acids and recycled components. Ubiquitin-proteasome, short peptides plus antimicrobial peptides. Mitophagy, complete mitochondrial destruction. Clinical links: autophagy, aging, cancer. Ubiquitin-proteasome, multiple diseases, cancer therapy target. Mitophagy, Parkinson's disease. Screenshot this table. It's your revision cheat sheet. Let's tie this all together with the clinical relevance, because this is where cell biology becomes medicine. Parkinson's disease, As we discussed, mutations in PINK1 or Parkin impair mitophagy. Defective mitochondria accumulate in neurons, particularly in the substantia nigra, and this triggers neurodegeneration. This is one of the clearest genetic links between an intracellular mechanism and a neurological disease. Aging. Progressive dysfunction of autophagy is believed to be one of the drivers of aging. As we get older, our cells become less efficient at clearing damaged organelles. This leads to accumulation of cellular debris and contributes to age-related decline. Cancer. The Ubiquitin-Proteasome Pathway degrades cyclins, the proteins that drive the cell cycle. Disruption of this pathway can lead to uncontrolled cell division. This is why proteasome inhibitors like bortezomib are used in cancer treatment, particularly in multiple myeloma. Antibiotic resistance. And finally, the most exciting frontier. If 92% of human proteins contain hidden antimicrobial sequences that the proteasome can release, this could represent a completely new class of natural antibiotics, one that bacteria haven't had billions of years to develop resistance against. This is the implication of the Goldberg et al. 2025 paper, and it's being watched closely by the scientific community. Alright, let's bring this all home. Today we covered 3 intracellular proteolytic mechanisms. 1. Autophagy. The cell's bulk self-cleaning system. Phagophore forms, becomes an autophagosome, fuses with a lysosome, degrades the contents. Failure leads to aging. 2. Ubiquitin-Proteasome Pathway. A targeted protein degradation system using E1, E2, E3 enzymes and the 26S proteasome. And now we know, it also makes antimicrobial peptides, as shown by Goldberg et al. In Nature 2025. 3. Mitophagy. PINK1 and Parkin specifically eliminate damaged mitochondria through the proteasome system. Mutations cause Parkinson's disease. These 3 systems work together to maintain cellular homeostasis, and when they fail, disease follows. If you found this video helpful, please like, share, and subscribe to Gray's Gyan, and hit that notification bell so you never miss a video. Drop any questions in the comments below. I read every single one.